expression and protein interaction approach to identify autism-rich genes. I'm an OmicsLogic fellow and a data science intern at Columbia University. Firstly, there is a high prevalence of autism, as 1 in 44 children are afflicted by the disorder. The clinical diagnosis of autism has been limited by the lack of conclusive biomarkers and the high genetic and clinical heterogeneity in regards to autism's etiology and hallmark symptoms. For CHD7, a high confidence gene, and SLIT1, one of the candidate genes I identified in this study, you can see that these genes share high expression levels and similar expression profiling. However, SDF4, a non-associated gene, has little to no significant expression.
Hello, my name's Nambira, and I have done a project on developing novel drug candidates for MDD, MMD, and hypertension, and I am part high schooler at Crystal Springs Upland School. So the second messenger cGMP is an extremely important target for disorders such as heart failure, atherosclerosis, and cardiac hypertrophy, disorders affecting millions. The SGC protein is a site that converts GTP to cGMP, a small molecule controlling vasodilation. Currently, very few to no therapeutics target the SGC-cGMP pathway due to many unknowns about the protein structure and qualities. It has a role in rare diseases like MMD, and it is extremely important to target cGMP to find treatments. cGMP can not only treat cardiovascular disorders causing up to 31% of deaths worldwide, but also have a role in cancer regulation, major depressive disorder, and more. For the genetic analysis, RNF213 gene co-expression, HLZ2 has a Spearman's correlation of 0.769. It is a co-actuator for the peroxisome proliferated activated receptor alpha. And OAS3 regulates the expression of chemokines and interferon responsive genes. Similarly, for the gene GUA1B1, MAP9 has a Spearman's correlation of 0.746 and is required for spindle function. So after the genetic analysis and structural analysis are done, binding affinities are collected through docking, molecular docking. So the 5 ligands with the topmost armistine binding values have their pharmacokinetic properties analyzed, and their chemical moieties were analyzed in pi-mole to reinforce if they were good candidates. By gathering this important structural information, drug candidates are proposed after conducting a virtual screening of thousands of ligands. Drug candidates, namely C15H24N2O2, C9H11NO, and C16H20N404, were identified. The drug targets identified can move on to lab testing on animal models to be developed further into medications that are lack of FDA-approved treatments. Finally, a combination of invasive treatment and medication can be used to reduce the chance of stroke or high blood pressure. A soluble guanylyl cyclase contributes to a variety of conditions from hypertension and MMD to even cancer. Further research in drugs targeting SGC would be imperative. So here are my references, and thank you so much.
So, hi everyone, I'm Rutvi Vaja, and I'm currently an undergraduate student in the field of biomedical sciences and research. Today, I would be presenting my research on gene expression meta-analysis of colorectal cancer tumor cells that reveals genes in association with tumorigenesis. Yeah, to start with the introduction, more than 12 million people are every day diagnosed with colorectal cancer and 600,000 people die from it. It's a very deadly disease. However, the pathogenesis of colorectal cancer is still not known. This work identifies 13 gene signatures representing 4 CRC tumor gene signatures and 10 other glandular tumor gene signatures that are colonic by origin. Gene set enrichment analysis approach have been used to define positive and negative CRC gene panels. From GSEA identified leading edge genes. The leading edge genes were identified and later used to verify enrichment and significant edge gene membership of CRC panels in different types of glandular tumors in origin. GSEA-based approaches here used to analyze the diseases and mRNA expression data of tumor and normal colorectal cancer tissues derived from human colonic biopsy samples by defining and comparing gene expression signatures. In order to examine the gene expression changes associated with tumor formation, in CRC, differential gene expression analysis was carried on using Welch t-test 2-sample analysis. The t-test score of clean and normalized value was used in order to define the gene panels. The t-test scores later defined 13 gene signatures which were of statistical significance. The signatures are formed from the gene list ranked from high to low differential gene expression between tumor versus normal samples. The signatures derived from the same data set use the same control samples for cancer. However, our work that identified certain kinds of solute carrier family genes could be useful, and it needs to be proved by wet lab research or Western blot techniques or qRT-PCR techniques in order to validate that these genes are involved in colorectal cancer disease tumorigenesis, which could assess that the GSE-based approach that we used is more useful in identifying certain kinds of genes, which normal bioinformatics tools like fail or are not that efficient to derive. So thank you so much.
So I have really enjoyed looking at all of the presentations. I've also really enjoyed listening to everybody today. I must say I'm quite impressed at the caliber of the presentations, but also of the work. And it's really great to see some early career researchers really tackling some real world problems. And that's been very exciting. All of the talks today did some hectic analyses, and I think you should all pat yourselves on the back. And I think you should also be very, very proud of your progress in such a short span of time. It's been truly impressive, and I'm really, really enjoying it. And I hope to be here for a lot more of today's session, so that I can hear some more from everybody else. But it's been really, really wonderful, and I've really been liking the posters as well. The posters are fantastic. So just well done, guys. It's really good.